Hey everyone, today we're going to be looking at the scientific method. Don't forget you have a graphic organizer which follows along these lecture slides pretty well. So let's go ahead and get started. In the world of the scientific method, scientists have to follow this specific pathway, this process, whenever they go about experimentation. The scientific method specifically is a series of steps that scientists follow to standardize the process of experimentation. It's really important that we have this process so that way scientists across the globe follow these specific rules every single time they go about performing some sort of an experiment, whether this be chemistry, biology, physics, um, AP environmental science, or rather just environmental science, stuff like that. This pathway is very cyclical, meaning that when we start the process, we want to begin obviously with making some sort of observation, but after you go through your experimentation, you always end back here at making an observation, or rather refining your hypothesis. We're going to go over each one of these specific steps in detail, but just to kind of begin, kind of remind ourselves the steps of the scientific method, it's going to be to make an observation. That observation is going to either be qualitative or quantitative. After we make a observation, you want to propose a hypothesis. Hypotheses can be like if-then statements, but really at the end of the day, a hypothesis is trying to help explain your observed phenomenon, the thing that you saw in nature that you're trying to explain. Why is it happening the way that it's happening? After we do that, you're going to design an experiment. Specifically, it's going to be a controlled experiment. Experimentation and pathways with how you can test your hypothesis can vary slightly depending on the field that you're in. But generally speaking, and what's true with all experiments is that you want to have a controlled experiment. A controlled experiment is going to be one where only one of the factors or variables of your experimentation is changed at a time. You want to analyze the data. Oftentimes, this is going to be done graphically in the world of chemistry. And last but not least, you want to make a conclusion. You want to either see if your analysis and data supports your hypothesis or refutes it. And one of my favorite things to kind of remind students is that we can never really prove a hypothesis to be true. You can only really support it. We as humans, we're only so good at scientific practice, and we might not have seen all of the variables that are actually going into affecting that science, affecting that phenomenon in nature. So with that being said, let's go ahead and start with observations. Remember, there are two types of observations. We have qualitative and quantitative. A quantitative observation is going to be any type of observation with measurements. And a measurement specifically is a number with units. You can take your ruler out, measure how long something is. You can bring your beaker into class and fill it up with some volume of liquid. As long as you say 25 inches or 35 milliliters, once again, a number with a unit classifies some sort of measurement, which is a quantitative observation. Qualitative observations, remember, are going to be any observation without a number. So this could be color, could be consistency. If we're looking at like a substance in nature, we're like, hey, that's a solid. That basically just tells us, hey, the particles in this are not flowing past one another. But notice there was no measurement with me claiming that that substance was a solid. Moving on, hypotheses, once again, are some sort of proposed explanation for why something happens the way it does in nature, in the world, right? And one of my favorite things that I had a very young student tell me one time, I think this is an elementary school student, he was like, hey, a hypothesis is a guess you can test. And he's not wrong, right? Like you're basically making some sort of guess, and then you're seeing if the guess you made is true, if it supports that hypothesis. Once again, it's a testable explanation for an observed phenomenon. And one of the last things that I love to kind of add in this part is that a hypothesis can't be proven true. It's that we always support our hypothesis. And this is what I was this is what I was saying earlier in regards to scientific practice and us just being humans. We can't always observe and see every single aspect of a scientific experimentation. So we give ourselves a little bit of room for error just in case we're wrong. Now, if we test a hypothesis over and over and over and over and over again, and every time we do test it, it supports the hypothesis, then eventually that hypothesis can turn into a theory, which we'll talk about later. So after we have proposed our hypothesis, we move on to a controlled experiment. Once again, a controlled experiment is going to be an experiment where only one factor of the reaction is changed at a time. Only one variable is changed at a time. Now, there's a couple vocab words that are associated with controlled experiments, which you might already be comfortable with, but it's good to review these things. The independent variable is going to be the factor of the experiment that the scientists change. It's the one that you change. We're saying, hey, I think that this chemical reaction happens at this temperature, but not that temperature. So you run the reaction at 25 degrees Celsius and then again at 75 degrees Celsius. The temperature would be the independent variable. The dependent variable is going to be, is going to change based on the independent variable. So as far as the example that I was just using, the chemical reaction running versus not running would be that dependent variable. It either occurred or did not occur, depending on the temperature with which we ran the reaction at. Last but not least, because we are, on planet Earth in a world where there's a lot of factors that could affect a chemical reaction or any sort of experimentation, we also have to make sure we understand that the control variables, the other parts of the system, must stay the same in order for our controlled experiment to be controlled. Controlled variables are, once again, they remain the same. And as far as the chemical reaction example I was using, we would want to make sure that we use the same amount of reactants or reactants to make the same amount of products. We would want to make sure that the atmospheric pressure stayed relatively constant when we ran that reaction. Same glassware, same experimental apparatus while we go through that particular experiment. Once we collect our data, it's quite important how we actually understand and analyze it. And as far as this goes, there's a bunch of ways that we can analyze it. One of the ways that we will often look at data in the world of chemistry is by graphing. When we graph data for a collected controlled experiment, the independent variable is always graphed on the x-axis, and the dependent variable, if it's quantifiable, is going to be graphed on the y-axis. This is a pretty much standardized way of doing this, and it helps us see the data in a really important, nice way. So last but not least, when we go through and are looking at our data, looking at the analysis, looking at the graphs, we want to understand that the data will either support or refute the hypothesis. Regardless of the results, you should retest the hypothesis to further define or support your hypothesis, further manipulate and change, tweak, and just kind of see what other things can change the experimentation. Or if it refuted your hypothesis, you want to change your hypothesis and maybe see how else you could change or affect that. So this was the lecture on the scientific method. Don't forget, there is the graphic organizer, which follows along with those slides. Thank you so much.